Here we go. Yes, it's starting. So hi, everyone. Welcome to the seventh session, the seventh global community call for this Open COVID-19 initiative. It's already the seventh time we gather together. It's been more than 6 weeks that we started this initiative, and wow, I mean, it's growing beautifully, not in terms only of size, but really of structuration. The quality of the project that you guys have been working on is really, really, really amazing. And we are going to go through what the current project have for updates. And also big news is that we have finally closed our first round of applications and for funding. And we'll be hearing about the project that won this first phase, this first round. The second round is also open, so it's not too late. The deadline is going to be on Sunday. So if you want to apply with your projects, it's happening now. But before we actually go through the projects, a few updates. As you know, we've been trying with JOGL to really put together, like to find as much help, institutional help as possible to support you guys in your projects. For example, thanks to the AXA Research Fund, we actually can fund microgrants. And thanks to APHP, we have also the ability to take some of the solutions that you guys have been working on and to test them in a hospital environment, etc. And so we've been working also on doing some more partnerships that can increase the overall value, of this program. And we've been doing this also with hackathons, like the Hack from Home hackathon that happened a couple of weeks ago in the UK. And actually, 3 of the best projects from that hackathons are now joining the Open COVID-19 community and will be supporting those projects. Unfortunately, they won't be able to speak at this session right now. It was just happening in not a coordinated fashion, but they'll be there next week to tell us what they've been working on. Yeah, so one thing I want to announce is that we actually got more support. So just recently, we made a partnership with the Vinci Foundation, who is going to support this program with 10,000 euros. So that's more for funding more projects. So that's great. And also, we've been working on how to extend this program in Africa. And so we've been working with many partners, local partners, to see how we could organize the same thing we've done so far here, but to see how we could, some of the existing projects that we've been working on could be forked, adapted in African context, and how also African communities can also join in the effort and provide their own solutions also to this crisis. And so we got support from AFD, which is Agence Française de Développement, or the French Development Agency. So it's one of the main public body to support the development in the South countries. And thanks to them, we'll be able actually to support projects that are either done by African communities and African partners, or that are targeting African-specific problems. So you'll be hearing more from that in the next few days. But that's very, very, very exciting for the global community here. The next part is obviously the most exciting one. It's the microgrants, the JOGL microgrants that have been going on. So the first wave is now closed, since all the reviews have been in. We got more than 40 reviews, so that's great. And I will call also for Chris and Elliot, who has been doing an amazing work on analyzing all the reviews that you've done, in order for us to really be able to have a comprehensive look at how good, impactful, or feasible the projects are, in order to choose the best projects. So JOGL will actually fund after this first phase 7 very promising projects, and which make a total amount of 15,000 euros in terms of funding for this first round. And the second round is now open since also last Monday. So before actually asking the winning projects to have a very, very, very short pitch, like 1-2 minutes maximum to remind the community here what they've been working on. I'm going to ask Chris and Elliot if they could present, their methodology for doing the review analysis, which, by the way, you found also as a PDF in the announcement channel and the project review channel, so you can actually have a look, like better look to it. So I don't know if Chris, are you here? Or Elliot, I don't know.
Hello.
Yeah, Elliot. Hi.
Hello. Oh.
So
Okay.
thank you so much for all your work. Do you think that you could maybe, I don't know if like share your screen or, yes, that's fantastic.
Chris has got it sorted.
Excellent, excellent.
There we go. Thanks, Chris.
Can you hear me now?
Yes.
Sorry? Okay. Well, basically, the methodology is, we did this as an open review. So you submitted your applications, and then anyone in the community could review the project, and they were scored for feasibility. So how easy is it to pull off your project? They were scored for how much money does it take to fund this project, and also they were scored for the impact of the project. So anyone could have reviewed your projects. You couldn't review your own project; that would be removed. And in the end, this is sort of the analysis we got. We did lots of other analysis as well, and that can be found in the PDF, but for time, I think this is the easiest way to show it. If it had a score below 3.5, and the score, by the way, were between 0 and 5, so 5 would be the best, 0 would be the worst, and it would be too weak, then therefore we wouldn't fund it. And then if it was above this score, like many of the applications you're about to see today, then they were funded. And they also weren't funded if it wasn't an R&D project. And I tell you what, like, all the projects were incredible, and thanks a lot for all the reviewers for reading through all this as well. Yeah, it was quite simple, really. Just review the projects, find the average review score, and then a meeting at the end to see which are the best projects and see the ins and outs of each one. And if you want more, thank you so much, Chris. So as you see, we have 2 projects in the area where normally they would be eligible for grants that we removed because they were not R&D focused, but more manufacturing focused, meaning like they were looking for funds to manufacture an existing object or device, which is not eligible for a grant, as we focus more on R&D here and research. But overall, yeah, it's been really fantastic. So now I'm going to call for the project. Can you actually leave the slide on so that? Oh, okay. Yeah, sorry. So I'm going to, who would like to speak first? Maybe we can take the let's see who was the first on the list. The cough check app. Do we have Hernan here? Oh. So maybe Hernan didn't make it. Okay, so then we are going to, can you go back to, Chris, can you go back to the main? Yeah. Yes, thank you. So Breezy, do we have Adam?
Yes. Hello.
Thank you, Adam. Congrats for your projects. Could you explain us like very fast, like 1-2 minutes maximum, what you're working on?
Yeah, thank you. So I'm Adam Krovina from Slovakia, and we are running an open source project of a ventilator, the mechanical ventilator for clinical use. More teams are trying to do this over the world. So we are another project, but we are also willing to cooperate with other projects because we have some ideas which I haven't seen yet, but we would like to spread them. That the ventilator should be run on the microcontroller with a small display, which would be cheap and it would be a stable system thanks to its simplicity. And we have, thanks to Bill Foot from California, we have an Android application in development which can be connected to such a system, receive messages being sent periodically from the microcontroller, and it shows nice graphs of online data of airflow, air pressure, and tidal volume changes, which are very normal to see on commercial ventilators. So I think this could be really helpful, maybe also to other projects. So we are also trying to contact team leaders from other projects and offer them our ideas and our software. So this is what we've been doing until now. Now, since we won the grant, so we can buy more parts and start experimenting with real hardware. I would say start building the ventilator itself as the whole device. So thank you.
Fantastic. Thank you so much, Adam, for all your work. I think it's fun. You presented the philosophy of this community very well, where the idea is not necessarily to work on your own project, but to actually look at how you can contribute to a more global effort. That's fantastic. The next project is the bioreactor project. Do we have Adrian here? Maybe Adrian didn't see the call. So next we have the project optimizing the NEB lamp test. Who would like to speak for this project? I think it's going to be Ellen or anyone involved in this project.
Sarah, I want you to take it, Sarah, because you're farther along than I am. Okay.
Okay. Sorry, I didn't know you were on here. I'm going to let you just go. Yeah. So the main idea with our project is to reproduce across several labs and optimize an NEB lamp test for testing for SARS-CoV-2. There's a publication with some primers and a protocol that is linked out directly from the NEB website, where you buy this this kit. So right now, 3 of us labs have been.
Hi, Sarah. Sorry to interrupt you, but your speaker is a bit difficult to hear.
Yeah, I think. Okay. Yeah, your connection is not very stable. Maybe Ellen, you can maybe pitch in. I don't know.
Yeah, okay. Sure. I can do that.
Yeah.
Is my speaker okay?
Yeah, it is.
Okay.
Yeah. Thank you.
Yeah.
Thank you for that, Sarah.
Well, Sarah and Izzy are both on, and they're both in the same room, and maybe there is some feedback or something weird. I don't know. Anyway, yeah. So as Sarah was saying, we started with this protocol, LAMP being this really simple diagnostic test that requires a lot less equipment than some of the PCR-based tests. And so it also, there's a version with a simple colorimetric output. And there's another group working on a version with a different output that Rachel will probably talk about later. But we started with a paper that was out of New England Biolabs that said that that it was a relatively sensitive diagnostic test that could be done easily. It can be done basically in a cup of hot water. So what we've been mainly doing is coordinating. Right now, I think the only lab work has been done across 3 labs, me and Sarah and Izzy's lab, and then Chris Monaco at the CDC. And we've done some preliminary tests. But the other thing is that there are a lot of people that are jumping on LAMP, and we're finding that the LAMP uses a primer set like PCR, except it's more complicated. It's something like 6 different primers. So we've been looking at other primer sets. So in some ways, we've deviated a little from the original title of it. but basically we want to optimize a colorimetric lamp test as best we can, whether it's changing the primers, looking at different viral transport media, and if they interfere with it, et cetera, et cetera, et cetera. So that I'm not sure how much else, Sarah, do you want to add something?
Can you hear me?
Yeah.
Okay. So the only thing to add is not super, I mean, the interesting thing is that we are able to detect the positive control plasmid that we're using. Where we're at right now, though, is still trying to reproduce across labs. So we're in the beginning. Sorry. And so that's kind of, we'll have more information, I guess, next week as we try the different primer sets and things. But we are able to detect the positive control. In our lab, we just received this morning the heat inactivated actual RNA virus. So it'll be interesting to use that instead of just the DNA and gene positive control and see what's happening in our reactions.
Fantastic. Thank you so much to both of you. It's very exciting. The next project is a Do It Together SARS-CoV-2 detective, which is also very much connected to the optimizing the NEB lamp test project. Who would like to speak for this project? Maybe Rachel? Yes.
Yeah. So hi, thanks to everyone for thinking this was worth funding. As an open public lab, for us, we're used to only having members fees for use of the lab. And so this is really nice to be able to go through and get some of these sort of cutting edge reagents and so on. So most of the last couple days since we heard the good news, we've mainly been looking into ordering things. And so, of course, here in Europe, we have to get some things, as we're a sister project, I would say, to the NEB lamp test project. We've really been trying to follow along with the NEB enzymes to start with. And we also have very good experience from the GMO detective example that we're trying to make a test called, we're calling it Corona Detective, in analogy to this test for seeing if there are transgenic genes in food products, which Guy developed in the CRE. So Guy is another partner in this project. And so we have one lab in Madrid with Fran, one lab in France, where Guy also has all the robotics and freeze drying equipment to make these tube strips that have all the reagents, including the control tubes. And what we want to do is choose the best set of primers that are really optimized and similar, of course, to what Sarah and Ellen were telling you about, to make something that we can ship to our other partners who are as far away as in Chile and in Cameroon. I saw that Tomas Mboa is here also, though I don't know if our friend from Chile, Fernan, is here also. But we're really looking forward to testing things. The good news is that I finally got an offer that seems like we can live with it. So we're going to be getting the enzymes. We're going to test against also the old school enzyme, which is more likely to be available as an open source version sometimes. So we can see if we can optimize also to not have to use something that's proprietary. And we're looking forward to getting into the lab. Even Guy has heard some good news that maybe he can even go into the lab before the 10th of May.
That's great. That's great news. All right. Thank you so much, Rachel. The next project we want to hear about is the COVID Alert project. So do we have Ali here?
I'm here. Can you hear me?
Yes. Hi.
So another lamp-based project for detection. You know, sister project, especially with the COVID-2 detective Do It Together. We are attempting to package reaction into a low melting point wax-driven cartridge with the ultimate goal of self-testing by people in their home or wherever they might be. and we're making steady progress. We've screened 3 different SARS-CoV-2 specific LAMP primer sets. We're packaging human mRNA specific control primer into our cartridge, and we're on like the 3rd candidate right now, and there's some exciting developments there. Our kind of main hurdle has been to get access to clinical samples to validate our primer sets. Everything works fine with the various controls that we can purchase. So we've been making progress with that, and hopefully soon we'll have some clinical samples to work with. Yeah, I'm looking forward to updating everybody on the progress of the project. And we have a little team here in the Bay Area. We're based in Oakland, California, that came together through the Center for Emerging and Neglected Diseases hackathon that happened a few weeks ago. So we have a little like self-selected group of public health people and other scientists who are working on it. So that's our brief report back.
Ali, can I ask a question?
Of course.
What do you guys have a lab in Oakland also?
Yeah, we have
where do you work out of?
Oakland Genomics Center.
Oh, okay, cool.
Who is this?
Oh, yeah, my name is Antonio Lam. So I'm a molecular biologist, but I'm based in Basel, Switzerland. I'm currently working on an app as well, but it's I can share that later, but I'm just
we should
learning
connect.
more about the projects. Yeah, but I'd love to connect with you as well.
Cool.
Okay, awesome. So what is great about thank you so much, Ali, again. What is great is that all those projects are so synergistic, and so they're all playing a different part, so that at the end we are making sure that we have the highest chance possible to really have a working protocol which is optimized, that can work as much for large-scale testing as much as individual testing. And for example, Ali's been focusing on using a cartridge technology to send this test at home. That's great. And the fourth diagnostic focused project is the cell-free system for sequence-specific sensing of SARS-CoV-2. Who would like to speak about this project? Maybe Fran?
Yes, thank you, Thomas. Well, first, thank you to everyone who have reviewed the projects, and thank you for the support. During this project, we started this 3 persons, one Pesta from Canada, Aníbal from Chile, and me from Madrid. The idea is to use cell-free systems, which are genetic modified organisms, but we will just explode the organisms, so we are not using living organisms, just the things inside of them, to produce colorimetric or fluorescent proteins in response to the presence of coronavirus. So what we are working on these weeks is in analyzing a few thousand of the strains of coronavirus to design the primers and the sequence that will create this colorimetric response. And also we are now that we have been granted, we are working with the quotations of all the products, getting all the prices, the list of materials. And also we are happy to see more and more people interested in this project, joining it. For example, Rachel from Switzerland, and also people from other parts of Europe. And that's all. I think we are just doing the list of regions now and analyzing the genome to design the sequence that we are going to use.
Awesome. Thank you so much, Fran. So as you can see, we had 4 diagnostic-related projects that were funded, and then 3 who were, so 1 for medical devices and 1 for diagnostics, which is the Cough Check app. By the way, so as an introduction to this project, it's an app with working on the machine learning approach to listen to your cough sound and see if the cough sound would have a signature which it would be COVID-19 related. And so would be a way to also, prevent or diagnose your the presence of the COVID-19 in your body. So that's great. One thing I have to say is the project whose applications were too weak for this round can still apply again for the next rounds. It's not closed at all. In fact, it's this review process is not only actually for asking money, it's also to be able to get reviewed by the community and get feedbacks. So it could be useful for you to just improve the overall quality of your application and project. So you could use this for that. The next round is going to end on Sunday. We will need reviewers again, starting Monday, to review the projects. So look out for the announcement channel. We'll be asking you to join. And yeah, I think that's it for the grant today. We're going to go to the next part of this call, which is going to be the usual weekly update from Mark about the status of the community and its dynamic. Mark, would you like to take the lead?
I feel like in a weather forecast situation. Hello, everyone.
What's happening in Europe or here in South America?
Actually, and it makes sense because I actually have a world map to begin with.
What's the weather like? Please.
So, do you see my screen?
Not yet.
Oh, you don't see it. Let me share screen. Desktop share. All right. Do you see now? Okay, yes, I was actually showing the world map for the weather forecast, So maybe a little update. So what we like to look at is a bit, where are the people from in the community? How did they arrive in the community and what's happening then? So in the meta-study group, first we look at the very macroscopic features. So here you have as you see that we still have a very broad reach of the community across the globe. We have many new visitors coming every day. So there's 500 people coming every day to check a bit the project. We have 20 new people registering every day to the platform. There's some new countries popping up here, like Italy, for example, the past week. How did they arrive? So another interesting thing is, how did people know about the community and arrive on the platform to check the program? So there's many just direct connection to the program page or to some of the project pages, but there's also some reach from different places where either through the press, whether it's Italian press or French press, whether from maker websites or even from GitHub repositories where people have put the codes of the apps of some of the projects, right? And that allows to redirect to the platform. And I would say there's a small number from the social media, and that will certainly get strengthened with all the accounts. And in the Google Doc, you can check all the OpenCovid accounts and retweet and reshare when you see posts, and it will help bring that up a bit. So just to show, I was very happy to see that we are in Sciences et Avenir, which is, my childhood French press reading. But we've been featured in as a community in some articles, some of which I had no idea. Actually, I just checked on the platform in Italy. So in terms of skill set, so the people coming.
Did we get actually featured by Gaël Giraud?
You know this person?
I mean, if it's Gaël Giraud, the past head of AFD and one of the best economists in France, yes. But maybe not. Maybe it's a different Gaël Giraud. I don't know.
Well, that's strange. Okay, we need to pin that down. I don't know. It's in an Italian newspaper.
Okay.
That's a weird serendipity. So we're also you know, and I've been promising that for the past 2 weeks, but we're still building the recommender system, and there's still probably 7-10 days more work to do on having that installed on the platform so that we can have a good matchmaking algorithm between the skills or the needs and the skill that they need and the users that come. And you know, we've been saying, for example, in the needs, some skills related to lawyers or some skills related to some coding abilities and or some, you know, web development, data engineering, project management skills. These are things that we need to bring to the users that can help on those needs. And we're working still on that. And we have a regular meeting on Mondays on that. So ping me if you want to know more. Slack activity is really stable. We have more new members. We're 1,100 now on the Slack. It's pretty stable, which means that the new members don't add up, right? They don't come and interact more with the base. So we need to have an effort still on the onboarding space. But at least it's stable, which is good. In terms of the different channels on the Slack, we represent them here by their importance in terms of number of posts. And you've seen an evolution with 2 projects in the past 2 weeks that have kind of been very active on the Slack, the syringe pump and the hack coronavirus. So I don't know if somebody will be presenting that later on. But so that's to give you an overview a bit of the evolution of the activity. And to get that better, we're just finalized, it's almost finalized, to put a platform for having a dashboard to visualize the initiative, whether it's the heartbeat of the channel, so their temporal evolution, or the community. It's an open source platform, meaning that we can also modify what we're going to display. And we'll be able to see the evolution in time of the different channels, to be able to cluster people according to different algorithms. So right now we're working on having a stable version of that dashboard. And after that, you'll be able to kind of explore more by yourself the Slack data. Voila. We still have a Friday meeting to discuss more about that. And there are some regular meetup on the sub teams as well to discuss this thing. And I'll talk a bit about the data challenge later maybe, but we're working on partnerships as well about data. I have a slide on the Google slide on that, so maybe I'll wait for later to give an update on that. But that's it for this one.
All right. Thank you so much, Mark. Next is we're going to hear from Leo. If there is any updates to share regarding the platform development.
Yeah, sure. So, I mean, there's just a few updates, which is that the main work that we had been doing sort of as a submarine, you know, which was to refactorize the front end and the back end to make it ready to have a much faster new feature pace in the future, is almost coming to a close. With we're going to start the testing phase tonight. So that's good news, at least for us. It was invisible to you guys, but it will come to light very soon, especially because the UX design that we're redoing also is progressing really fast with some very new features that are gonna come up, I believe, next week, very likely. So we'll have more news on this very soon, and you'll be able to enjoy a much better user experience overall. We've been working also really hard at getting this recommendation system that Mark was talking about between like needs and users. And so like the backend architecture is almost ready now. We are basically going to like start to enter the testing phase as well, you know, kind of soon. And so basically coming soon is going to be a major redesign of the program page at the program level, so that you'll be able to access much better, like much more up to date and better information flows, so that everybody can stay up to date in a much more easy manner. We should have better social media capability. That also comes into what Mark was saying in terms of like, you know, you guys resharing and things like this on social media. It'll be easier for everybody to do that. very soon we'll have notifications system as well, and so that's going to be, I think, very important for everybody, because a lot of people asked us to have this sort of feature so that they can know what has been happening since the last time they connected, and to come in perspective, and I'll chat about it with Mark when Mark speaks about maybe the data challenge, or maybe not, but basically we're implementing a version of data lake-ish on JOGL. So instead of hosting directly, I mean, you'll still be able to do that, but we believe that it is more important for all of you to be able to add external data sets to your projects, right? If you're working with Open Humans, for example, or if you're working on data that comes from a government that is hosting a CCAN server, such as data.gov or the European Open Data Lake, and you want to be able to integrate those data into your project, you will soon be able to do this. This is all coming within, like the next 10 days, basically. So a lot of new exciting features that are going to start popping up, and the rhythm is going to only accelerate. So don't hesitate to contact us if you have great ideas about things that we're clearly lacking and that could make your life much easier. Yes, I keep reiterating this message, but very seriously, if you have needs that can be covered by JOGL, tell us. We're here for that. Yeah, thank you.
Awesome. Thank you so much, Leo. I think Eve wants to ask a question. Wait. Here you go. You need to unmute yourself.
Yep. So hi, everyone. Just to make a teeny remark, I have already said this to Alex, is that it should be important to split the announcement on the channel announcement, to split the announcement of the project and the announcement of the new arrivals, new incoming people, because the visibility of the project is not strong enough actually on the platform. We have something like about 40 to 50 projects, and they are completely hidden by the thousands or 1,500 people arriving. And this is a pity.
So there are different ways to, you mean like you would like to hear more about the project updates? That's what you mean?
No, such that there is a location where people can find all the running project extremely quickly.
Oh,
do you
have a description of this running project very quickly?
So so you have to go on JOGL basically for that. That's where you see the different projects, and then you can actually look for project. Normally, actually, they put the channel or the lead. You can find the name of the leader of the project there. And then try to contact them either through JOGL or if they're on Slack directly.
Okay, just because at the end, what you would like is that the people arriving find most of the people arriving want to help.
Yeah.
So if they have opportunity to find very fast and very efficiently some project, the best will be the better.
Yeah. So I think it's something we need to improve grid here, and so as we said also, like the onboarding process can definitely be improved by Bonzo. So thank you, Eve, for the remark, and we'd be working on that.
Thank you very much. Yeah, and we're working really hard in UX right now to do exactly this. This is our #1 priority: is how do we get people that arrive or already have been here, and so that they can find where they can help the best, and to do this in the easiest manner for everybody. That's exactly our goal, and this is our #1 priority right now.
Mm-hmm.
And Leo, maybe this is a very nice transition to what the governance is trying to do with the on the Slack part. And just to answer your comment, Eve, I think both on JOGL side, we're trying to improve the platform to exactly like suit your need, but also on Slack, we're trying also, we're working very hard on onboarding, and and also synchronize everything so that your project gain visibility and it's easy for people to join it and to work with you. So maybe, I don't know, Thomas, has anyone or should I continue with the governance team as it was, or is there?
I'd like to go through the project first, and then we go to the working groups.
Okay. Yes. Sure. Okay.
All right. So thank you again, Eve. This is definitely something that we want to improve. We'll be working on that, and you're welcome actually to join also a group on the governance team to suggest some ideas if you want. And I've seen some ideas also directly on the chat, like we need actually a better connectivity between JOGL and Slack, and this is definitely a good point. So the next part of this call is to hear. So we've heard about the project that won the grant for the first round, but there are also many more projects that would like to provide some updates. So we usually every week we ask you to, if you're a project leader, to update a slide deck, with what has been going on with your project recently, so that we can have a look to it. So here I've put the list in the agenda of all the projects, and I'm going to call one by one the project and the people that are behind to provide updates. I won't be calling the project that we heard already about, except if they really want to provide like further information, let me know. So this time we have also Thomas Mini from OpenCell, who is here and couldn't be there, for example, last time. And he's been working with OpenCell on a way to do mass testing in the UK. Thomas, would you like to provide an update?
Yeah, absolute.
Thomas.
Hi there. Hey, can you hear me okay?
Yeah, absolutely.
Yeah. Great, great. Yeah, so I just wanted to share a bit about what we're up to. So it's a fairly big project. So I apologize if this takes a bit, but I suppose at the kind of high level, the idea is to have a modular laboratory design that can be easily replicated and sort of copied and sort of developed by other places around the world. So at the moment, I think the three pieces, if I could put it that way, are the first one is the laboratory, which is a shipping container, is what we have been working with due to its abundance, and also the fact that's really what we've been doing as OpenCell for a couple of years. So the shipping container really needs to comply with ISO guidelines alongside a couple of details about medical diagnostics. So there's a couple of ISO guides as well as UCAS certifications. I'm sure there's different ones in the US, but we really have all that on our webpage. So it's opencell.bio/coronavirus, and everything there. There's blueprints and everything like that. Everything there is actually really useful if you're looking to do that sort of lab building. Then sort of on the other side of it is the actual biological workflow. So how we do the testing. So the attitude we took here was we needed a kind of an open source, both device agnostic, consumable agnostic, and reagent agnostic workflow that doesn't put additional pressure on, for us, what's most important, NHS consumable pipelines. So if we were just using thermo reagent, the problem would be that we would basically be taking, the National Health Services here, reagents that they crucially need to be doing testing of their staff. So that wouldn't be a good idea. We've also noticed that there's been a huge amount in the media about shortage of reagents globally. While that is true, what really is the shortage of specifically vendor-locked consumable components for automation systems provided by Thermo Fisher, QIAGEN, and Roche. They're kind of the three biggest ones. This is a big issue. I'm very concerned about, people talk a lot about big pharma. Big bio is actually a lot eviler. They're really about to really consolidate even further. So you're about to see QIAGEN be purchased by Thermo Fisher. So I would speak to policymakers about that if I was you, and you know, if anyone in the EU is here, I mean, there should be an antitrust investigation there. It's an absolute disgrace. So really what the global supply chain challenge is that Thermo can't ramp up their production of the qPCR kits, as well as the MagMax magnetic bead extraction kits for doing high throughput RNA extraction. So we've really gone down a different route, which is using BOM Bio. So it's bio on magnetic beads, .bio. It's from a guy called Thomas, I forget his second name, who works in Cardiff University. He's kindly provided these reagents. So that's the process flow we wanted to use, something that anybody could use anywhere. And in principle, while it would require some fairly decent chemistry, because you've got to do this sort of silica loading of magnetic beads and, you know, magnetic nanoparticles you do have to buy, and you know, it just makes it easier because you're not putting pressure on those already strained supply chains. So that's kind of the molecular biology workflow that we thought about. So it needed to be consumable, reagent, and device agnostic. So we used an Opentrons to move the liquids around in these different wells of a 96 well plate. And we're working with 3 different PCR machines at the moment, so that people would be able to say, well, I've got that one, so I could download the protocol here. Or I've got this Opentrons and this is the particular system I'm using, so this would be my protocol. So it'd be a ready to go, you know, comparably easy process to sort of get started if you were in South Africa or you know, somewhere in the continent of South America, someone who has these facilities, you know, somewhere within their country, but really just needs to pull the pieces together. Then I think the final thing that I'd actually really kind of, this is my call to action for this call, is we've been working really hard on the software to be able to do a kind of a lab information management system. So it's often abbreviated to LIMS. These laboratory information management systems there's a diverse range of them, and it's a really interesting activity because you can build drivers for machines and integrate those machines in. And sadly, like most things in biology, every company that does it is closed source. And it's very unfortunate because actually it's a place where the open source community, particularly developers, if there's any developers on this call, could contribute enormously to the process flow and the progress of testing. Because really what we have is you know, a few innovators like Synthase who have opted to go down the closed source route. And there's very few options for just saying, I want to have an open source lab management software system. Now, I have actually gone through a lot of resources today, so I'm just going to save, if you'll give me 2 more minutes, I'm going to call out a few bits and pieces for people. So again, I'm going through the NHS a lot because for us, being able to integrate into that system is super important, because here medicine is provided through that single point of care. I don't know how it is in other different countries, but ultimately what is relevant for other different countries is there's a protocol called FHIR, which is, sorry, what it stands for. It stands for, I don't know, but it's basically a standard, an open source standard for healthcare data exchange. And it's really managed through a corporation called HL7. And realistically, what they do is they're kind of, you know, system agnostic, but what they've done is to have a set of standards in XML, JSON formats, where, and obviously sort of workflow formats as well. So ideological formats of how do you manage, let's say, patient to diagnostic to critical care to, you know, report test and all that. So really for us, what's most interesting is how do we integrate with a lab management system that's present in the NHS called WinPath? How do we collect data from that using an API? And there's a number of FHR APIs that are listed on the developer page of NHS. And how do we then have a compatible XML format with the appropriate details of that test, and then get it back into the system. So that kind of API portal, that sort of messaging portal is a little bit uncertain for us. We tried to contact the executives at WinPath. You know, they didn't like the idea of us sort of using the software, and I think that would sort of, you know, I mean, they made clear they have 75% market share of all GP surgeries across the UK, general practitioner surgeries. So they were pretty clear that they like their market dominance, and that's how they want to keep it. So they weren't hugely interested in us sort of helping. But I think that, you know, like any kind of, I mean, they were particularly evil, I have to say. I mean, like anybody, they're prepared to do business, and we could get a license off them or something like that. So I think if anyone on this call is interested from a developer perspective in working with particular data formats that we would need to integrate and interact through this FHIR standard, I'd be really excited to talk to you. As I say, if you want to reach out to the project, it's opencell.bio/coronavirus, and we've got everything there. You can drop us an email, you can call us. There's the blueprints if you are running a lab and you just want to copy and paste stuff. Yeah, there's lots more. So thanks for taking the time to listen, and thanks for the opportunity to speak, Tom. I really appreciate it.
Thank you so much, Thomas. This is a fantastic project, and it absolutely need to go through. So we'll do anything we can to support you. I think so. Yeah, definitely. If people on this call can help, but compared to the global community, this call is pretty small. I think what would be great is to make an announcement also in the Slack and on JOGL, and probably make sure also that you have a JOGL project page that people can actually just go there and know about what you're doing and what are your needs. That would be super helpful for you. So again, thank you so much, Thomas.
Thank you.
We're going to go. So the next project we want to hear about is the Edu game about COVID-19. I don't know if we have anyone in this call actually working on this project right now who would like to provide an update. I think Julie Legault is currently sick from COVID-19, what I've heard recently. So I wish her the best. So I will go to the next project, which is the COVID-19 Museum by Yves. Yves, would you like to provide an update?
So you hear me?
Yeah.
Yep. So I'm sorry, there is the phone ringing on the other side of the flat. But sorry. So the object of COVID-19 Museum is to collect all data arriving during this period of the COVID-19. So it's something huge. I would like just to let you imagine for 1 second. It's covering images, video, it's covering interviews, it's covering text, scientific text, or whatever. So what is important in this period is that it's not the first pandemic of the modern era, because Spanish flu was already a pandemic of the modern era. However, it's the first pandemic of the electronic era. And we have, for the first time, the opportunity to get all the information produced during this period and after this period on the topic COVID-19, recovered, to gather all this information and to offer the opportunity to not only look at this information later, but also to realize analysis on this amount of data. We have also the opportunity to explain later for the children. I'm thinking to small children which are today confined, children between 4 and 7 years old. I don't know if you have realized that they will be confined about 10% of their own life, which means that in some sense, they will consider that this COVID pandemic can occur with probability 1/10. It's huge. So this is the idea. The idea is really to build in real time, sorry, a museum made of all objects. So it's collaborative here, and it's for memory, it's for pedagogy, and also for analysis later.
This is the best way of using the virtual background of Zoom. Well done.
Yes, sorry, it was a little bit tricky. So when I have put the needs of the project, so first of all, I would like to say that UNESCO has sent a letter to all governments today to ask that official papers about COVID-19 crisis are kept for memory later. What I am thinking is that we don't need only official paper from the government, we need everything. Everything from this period to be collected. It will be useful for social science, it will be useful for health science, it will be useful for economy, it will be useful to protect the new world which should be created after this crisis. So naturally, with respect to the amount of money you have seen on the picture about reviewing that the amount of money asked for this project is huge. Indeed, I have included the amount of money need to only store the data per year if we are going on a private company. What we can hope and what you should hope is that such museum which will need copy in each country should be made by the government of your own country, and it should be protected inside the university, such that at the end, the price of a such museum is much less. We need people involved to code the website. We need people to code the database. It will be participative, not only later to collect things, but it will be collaborative from the beginning if you are involved in this project together. So you are welcome, and you can contact the project with contact@covid19.museum. Thanks.
And you're also on the Slack, right?
Yes, I'm also on the Slack.
Right. So COVID-19.
And also on JOGL.
Right.
It's project 175 on JOGL. And you will find the document there, you will find the contact, so you are welcome to join. I do agree that it's not a biological project. I do agree that it's not a health project, not a reacting project for medicine, but it will be useful for you for later.
Definitely.
Idea.
Thank you so much, Yves, and I completely agree. We started, focusing on the immediate need regarding the crisis, but more and more we are receiving demands and projects that are more focusing on the recovery, what's going to happen next. And I think your project fits very well into this. It may actually call for the opening also of a new challenge in this open COVID-19 crisis.
Yep.
All right, thank you so much, Yves, again. The next project we want to hear about, if the person is here, is the mass spec project with Nivi. Do we have Nivi here? Okay, I'm going to the next project. The mental health project with George. Okay, so we're going to the next project. Then we have the syringe pump project. Do we have anyone from this project? If not, oh, Sophie raised her hand. Do you want to speak, Sophie?
Yeah, hi, how are you? I just wanted to give you a heads up with George. He can't be on the call today, but as of yesterday, we're starting to work a little bit on this mental health initiative and getting some mental health support to people who may need it. A few topics that we are currently kind of playing around with, and we would like to offer weekly workshops and also a weekly open discussion. Some topics would include recognizing your new normal and staying flexible and pivoting, how physical organization can help a mindset, using sudden change and trauma to your benefits, how to elevate your energy, and finding value and power in living in the present. So if this is something you guys might be interested in, keep up with us and join our channel, and hopefully we'll get going within the next week or so. That's it.
Thank you so much, Sophie. Then for the syringe pump project, the open syringe pump project, do we have anyone from the project here? If not the case, I can speak for it. So 2 weeks ago, we got a demand from APHP, the Paris Hospital Association, that they were missing syringe pumps. It's a piece of machine which is not only about 3D parts, it has electronics, there is some fluidics inside, software, some software also for control for the nurse, for example. But just in APHP, there is a lack of 1,000 syringe pumps right now, for example. So we've launched a project 2 weeks ago, and now we have more than 60 people in the group, and the energy is just fabulous. It's been, I think we are only a week away from having a prototype being tested. We started from an open source syringe pump model that is called Poseidon, and we have the creator of the Poseidon directly in the group, who is now based in Caltech. So it's been really, really, really fast, and APHP has been also very much collaborative, providing us with all the specs, providing us also with reviews, feedbacks on the proposition we are making. So it's a beautiful collaboration, so we'll probably know more about it in next week. The next project we want to hear about is the enzyme production project from Isaac Larkin. Isaac, are you here?
Yeah. Hi, everybody. Can you hear me?
Yes.
Okay. So basically, we are trying to make it easy for people to manufacture their own enzymes for use in all sorts of diagnostic assays. So this is primarily a collaboration between the people who are involved in the Free Genes and the Open Enzyme projects in the United States and and Europe, respectively. The place we're at with it right now is we have obtained via donation and open material transfer agreement several expression vectors for enzymes that are commonly used in both RT-PCR and LAMP. So I think we have expression vectors for BST DNA polymerase, for a couple of reverse transcriptases, and what we're in the process of doing, right? And so those, I believe, can be distributed to people who want to experiment with manufacturing and purifying their own enzymes. And what we're in the process of doing right now is putting together a larger collection of many different diagnostic enzymes, along with many different possible purification tags, and submit that to Ginkgo Bioworks for synthesis. The idea will be we'll have essentially a library of both plasmids and parts that can be easily assembled into new plasmids to produce any enzymes that would be useful for any diagnostic assays, and to purify them in the simplest and cheapest way possible that's accessible to whatever resources you have available. So we are working on a JOGL funding application for that, for this project, and anyone who's interested in joining us and contributing is more than welcome to.
Fantastic. Thank you so much. I'm going to, do we have anyone here who would like to provide an update for a project? That would be the moment to do it. Otherwise, I'm going to go to the work group very fast and we'll be ending this, this call. So Rodrigo, you want to speak?
Yes, hello, can you hear me?
Yes.
Okay, I'm gonna share my screen 1 second. You tell me if you can see that, okay?
Yes.
All right. Sorry, 1 second. I'm having some issues with the privacy right now, so sorry for that.
Maybe we can go with John in the meantime, and then we can go back to you, Rodrigo.
Okay, cool. Thank you.
Yeah, John.
Yes. So John with the Epi Modeling Toolkit project. So a lot of things happening over the last week, and it is starting to seem like the name is maybe not the appropriate one. But most of the progress over the last week has been around doing customer interviews with epidemiologists and modelers around what their needs are. And a lot of the results of that have been causing me to pivot towards really focusing on building a open data exchange or knowledge base that epidemiological modelers could use to make sure that they have the best estimates of the parameters for their models. And so working on a lot of spec work there around how to best achieve the needs that they expressed, which include peer review of the papers that are used to create the parameter estimates, or the code that is used to create the parameter estimates, and various collaborative tools. So if anybody's aware of the idea of Blackboard systems, essentially the software design principle where a control system updates a knowledge base iteratively, and it can trigger reflows in a dependency graph where if different parameters depend on each other in some way, we can get information about whether the information conflicts or anything like that, is kind of the direction that we're going with this now. Beyond that, there has been, over the last week, quite a bit of work in doing a little bit of within-host mechanistic modeling. So I worked with a biostatistician to start to look at emergent properties of different kind of immune system responses within the same kind of mechanistic complex systems framework that is being used for some of the more epidemiological scale modeling. And there's a lot of work to be done there, but kind of the goal is to be able to come up with theses about, lysis or cytokines or, T cell activity or initial viral load or all these different things, and then understand how it might interact with the disease trajectory, not for the purpose of actually, modeling it accurately, but just having a better intuition about, more research that needs to be done. Okay. Beyond that, there's also some work on some communication related stuff, building kind of protocols for design of not academic papers, but just visualizations and other kind of explainers for lay people around different properties of this type of epidemic. So working with someone around kind of modeling social distancing as a network graph and understanding what happens if you were to contact an additional person or an additional two people. as well as doing things with looking at how artifacts of exponential growth can result in temporary plateaus or dips in the number of cases that may not actually mean that we've hit the peak. So lots of different projects going on, but the focus at this point is moving towards building a open data exchange that allows researchers to collaborate on iteratively improving their estimates of different parameters of the disease and of its reaction to infecting the public.
Thank you so much, John. Thank you so much for all the updates.
Sorry, really quick as well. Just wanted to mention that I'm also in contact with, there's another team called Corona Y, who's working on mostly on modeling the, with natural language processing, all of the papers that are being produced and building better tools on top of that. But as they've gotten organized, they've started to express interest in some of the developers there. There are about 800 developers and data scientists there looking at helping other projects. So Thomas, Mark, definitely want to connect you with some of the leadership there.
Definitely, that would be great. Thank you so much, John. Rodrigo, are you ready to share?
Yeah.
Like very fast because we're already burning the time right now.
Oh, no problem. So I think it's okay. I might leave this communication for the next meeting, and that's all, because I'm having some issues with sort of screen sharing and stuff.
Okay.
So that's fine.
Okay.
Thank you.
Okay, thank you so much. For next time then, Rodrigo.
Okay.
So very fast, what we're going to do right now, thank you so much again for staying up till now. This call is probably getting the longest so far since the beginning. So many news to share. We'll be talking about what were I'm going to stop your sharing. We're going to share what has been going on with the work group, like very few updates, the core updates. If you know, Camille, Alex, Hans, Pauline, and Chris, if you could really make it as short as possible, that would be fantastic. Maybe Camille wants to start with the governance working group.
Hey, sure. Okay, let's try to be very short. I'm just gonna share my screen quickly, and if it doesn't work, then I'll just stop. Okay. So as far as the, let's go. Yeah, so the governance team is about 100 people. I just want to tell you that it's open, and if you're interested in joining and focusing on facilitating the collaborative work and making it easier for everyone, you're very free to join us, and that would be a real pleasure. So what we do is that we create process, tools, frameworks so that people work more easily together. Just like the latest updates, we are currently working on improving the onboarding. If anyone is interested in improving the onboarding, making suggestions, please reach out to me. My contact is here, Camille Maslow, or you can also just come to the governance channel. We also are now holding ambassadors meeting, which is kind of a forum for everyone who has kind of a leadership on a working group or on a challenge, and this is where you can bring your pain points, and where we share together and we answer your pain points in terms of organization. So if you're interested in being an ambassador and joining these meetings and being more in touch with all the other groups, just reach out to me. For everyone who's in the community and working on Slack, I would like to recommend you to use Workstream for task management. This is something we've tried out and which works. And also would like to call for community member who would like to be more engaged in helping each other as moderators, guides, and recruiters. So I'm just gonna go very quick about it.
I think the people will have to go to the governance channel to learn more about that. If you want to get involved,
so that's the channel you can go to. If you'd like to be involved more in helping each other in the community, you go either to me or to this community support. And I'll be happy to talk about it more. giving the mic to, I guess, to Alex. Should I? I
Sure.
can continue sharing my screen if you want.
Yeah, that would be great.
Yeah. Sorry, you need to share it again.
Yeah, it's okay. No worries.
I'll talk very briefly as well. So quick update from the community management standpoint. We don't have a set channel for that. It's very cross-cutting. It's a very cross-cutting initiative. So I've talked to quite a few people in the last week. I'm conducting stakeholder interviews to learn more about project level teams, what's been working and what can we improve. So I have another 10 or so scheduled for the rest of the week. If you want to chat, you can click on the orange button if you have the link to these slides. Otherwise, just use that URL. There's still a few slots that if you want to chat with me this week and be part of this research project. Two quick announcements on my end. I want to spotlight the channel program question support feedback. If you have challenges within your program, your project team, you can share those here. And in that channel, there's a pain points Google Sheet where you can add your pain point to that as well. We check that to help address your issues. And then if you have specific suggestions on how the program can be run more smoothly, we created a new channel suggestions box. So it works just like a suggestions box. You just post your suggestion. And we'll be looking through that channel regularly as well. I'll be moderating both of those channels. That's it for me.
Thank you. Thank you. So next we want to hear about the communication team. Hans, are you here?
Yes. Hi, everyone. So a quick update from the communications team. Over the past week, our social media pages have started becoming more active. So importantly, if you have an account with Facebook, Twitter, Instagram, and or LinkedIn, can you please follow us? You'll find the links easily or just looking for OpenCovid19 on those platforms. If you engage with our posts, that is to say, put a like, comment, and share the post, then that will tell the platform that it's a good post and the platform will show it to more people. So please engage with our posts because that's definitely what we want. And the last thing is that you can now draft posts for our social media pages. You just have to make a Google Doc in the folder that's linked on the slide, and I will put the slide in the chat now. And once you've made this draft post in that folder, you can fill in the posting schedule sheet. The link is also there. And we will review the post and hopefully put it on our social media pages. And that's it for me. Thank you for listening.
Thank you so much, Hans. Next, we want to hear about the student engagement team with Pauline.
Hi, very quick. First of all, I just want to thank everybody who has joined in the past week. We are already at 50+ members, and that's really amazing. Important announcements we do, if you are looking to be someone who wants to reach out to other students or provide like a mentoring role or within a project reach out to students as a mentor, we have a mentoring survey now. The link is in the slide deck. Please fill that out. It will be super helpful to us going forward and developing the student engagement program. Also, spread the word. We have now been posted on all 4 of our social media platforms, so I will shortly be sharing the links to those posts to share in the student engagement channel. And also, if you haven't already, join #program student engagement and introduce yourself, whether you are a student or early career or you are a project and a program looking to reach out to a student. There are many students have put themselves out there on the channel, and I think they would love to connect. So once again, thank you so much for the interest and support so far. We intend fully on keeping this active and lively and a great resource for students, not just students, but just everyone in the community looking to connect with students and early career members in our group.
Fantastic. So much, Pauline, for all the work. So the next group we want to hear about, I think we've heard actually a lot about it in the beginning of this call, is a project and grant review group. So if you want to help with the review process and want to improve it, to make suggestions, there is a channel for that, which is open. Feel free to join it. And I think I will close the call actually now, but before we go, we have, I see that people are used to it, are already activating the video streams. So I will make a screen capture with everybody. So if you can activate your video streams that we can see you, I would ask everybody to say hi so that we're able to share this beautiful picture of the community to the social medias. So we are almost there. I don't know if Antonio or Sarah are on. All right. So ready? 1, 2, 3. Fantastic. Thank you so much again for being here. We're doing like great progress, and next week we'll have even more announcements to make. And I'm looking forward to see all next time on JOGL, the Slack, or during this call. Bye-bye.
Bye, thanks.